that the will work, and then what we'll do is we'll transcribe it, and what we'll do is we'll send it out to everyone via email, and we'll also post it to our website. So without further ado, I'd like to send it over to Dr. Jett and Dr. Spriggs.
Hey. So, good afternoon, everyone. Pretty exciting. We've got 100 registered participants. I'm Dr. David Jett. I'm director of the NIH CounterACT Program, and I'm joined by our program manager, Shardell Spriggs, today to present an R entitled Current NIH CounterACT Funding Opportunity Announcements, Web for Potential Applicants. So what we do is we will present the slides and then take questions as Tim and questions that were submitted or any new questions at the end of the webinar. So please mind that specific questions may be best done on a one-on-one basis with NIH staff at a later time. All right, Salty. This one? Little technical pause there. So, webinar goals are to provide a description of the NIH CounterACT program, talk some detail about our specific funding opportunities, and then to provide some tips and lessons learned for folks who are thinking about applying to the program. So, they've supported research since 2006, and its mission is to understand fundamental mechanisms of toxicity caused by these chemical threats and apply that knowledge to develop promising therapeutics. All goals are to improve the nation's medical response capabilities, support cutting-edge research, to improve our knowledge base, and optimize lead compounds for potential transition to advanced development. So that's the most common question we get is about chemical threats we need studied. Presented here are broad examples of chemical threats of interest, include traditional chemical warfare agents such as sarin and VX and Soman, chlorine, sulfur mustard, toxic industrial chemicals such as cyanide, hydrogen sulfide, and others, toxic agricultural chemicals such as insecticides like carb, chlorpyrifos, parathion, and rodenticides like rodenticomb and tetramine. Finally, we are interested in pharmaceutical-based agents, for instance, the opioids, fentanyls. And the program is guided by a list of about 200 chemicals developed by the federal government, and should contact myself or Dr. Spriggs to confirm that the chemical or chemicals they wish to study are on the list before developing an application. At this time, share that list with you, but we can discuss the list in detail with you to facilitate your choice or confirm your choice that it's on the list. What is helpful and is not included in our funding announcements is our list of toxidromes. This is, we have grouped chemicals on the list based on their mode of action. This gives you an idea of the kinds of chemical threats, but countermeasures developed for one chemical may also be effective for another chemical threat in that category. And these toxidromes include things like anticoagulants, rodenticomb, blood agents like hydrogen cyanide and sulfide, cholinergic warfare agents that we discussed, cholinergic pesticides like parathion, chlorpyrifos, and phorate, and aldecarb, like picotoxin, TETs, and strychnine, hemolytic and metabolic agents like arsenic trioxide, gallium sulfate, and arsine, opioids, the fentanyls, diacylmorphine, low pulmonary agents like chlorine and phosgene, and upper pulmonary agents like ammonia and sulfur dioxide. Finally, the vesicants like sulfur mustard and phosgene oxide. So, truly a trans-NIH effort because of the diversity of chemicals and their effects. Other NIH institutes and centers participated in the program based on expertise needed to cover ocular, skin, pulmonary, and other effects of the chemicals we studied. The National Institutes of Neurological Disorders and Stroke, Arthritis and Musculoskeletal and Skin Diseases, Environmental Health Sciences, Drug Abuse, Institute, and the National Institute of Allergy and Infectious Diseases that provides oversight and manages the contracts. IC has a pro officer assigned that can help you with specific questions, and rely on other ICs from time to time for advice, as the Institute of Child Health and Human Development, National Library of Medicine, and Heart, Lung, and Blood Institute. CounterACT program is designed to support research from the discovery phase, including the identification of therapeutic targets, the development of optimized lead candidates ready for advanced development by other agencies like the Biomedical Advanced Research and Development Authority, or BARDA, or other entities. The program includes several types of grant programs to be discussed later, and contract core facilities managed by the Chemical Measure Research Program at NINDS. These programs support grant projects and used to generate preliminary data for grant applications. The NIH-supported interagency agreements with the Department of Defense laboratories that conduct research in this area.
The programs that we will discuss are progressive. Start exploratory R21s through lead identification and optimization UO projects that result in candidate therapeutics and major advances in our general scientific knowledge base in the literature. It's important to read the funding opportunity announcements listed here and talk to the staff at NIH to see specifically where you may fit among these programs. Site to see latest funding announcements as well as a wealth of information including NIH scientific contacts, of funded investigators and publications, tips on writing milestones, courses, and other information. Again, it's critical to use the latest funding announcement when composing your application, since the entry requirements and review criteria may change from one version to the next. The R21 funding opportunity announcement is intended for exploratory or developmental translational research projects. They often serve as an entry point into the program for many investigators, including new or junior investigators with promising project ideas that have little to no preliminary data. These are sometimes referred to as high-risk, high-reward projects. Funded investigators often use this mechanism for follow-on studies that are out of scope of their project, such as validation in vulnerable populations. The goal of R21 is to generate preliminary preclinical safety and efficacy data to facilitate the development of competitive applications to more extensive support, such as the CounterACT UO1 mechanism. This includes, but is not limited to, the creation and validation of screening assays, animal model development, identification of therapeutic targets and biomarkers, and development of proof of principle efficacy data for candidate therapeutic compounds. Each funding opportunity announcement has one receipt date per year. Here is a schematic of the next funding cycle for the R21. The application receipt date is May 28th, with review in October, and awards will be released in March of next year. The action budget may not exceed $275,000 in direct costs for a 2-year project period. with more than $200,000 requested in direct costs in any single project year. Please keep this in mind when writing your application budget. One is not renewable. One receipt date a year, you will need to time your studies to make sure that you have enough preliminary data well before your grant runs out to apply for the next level of funding in the program. Ensure that you don't have a gap year in your funding. R21 in the example shown here, if you receive your award in March of 2020, you will need to apply for U01 funding 1.5 years into the award. I thought it was important to mention this here because we have so many R21 investigators that also apply for U01 funding.
Okay, so the U01s. So this is the next sort of level of research that we support. U01 supports research on the identification of lead compounds. So by the end of the 3-year project in this case, you should have a compound or compounds that is biologically active and synthetically feasible, where toxicity, affinity, potency, target selectivity, efficacy, and safety have been established both in vitro and in vivo models. Includes, but is not limited to, mechanistic research that leads to a lead compound in 3 years, combination of targets. It's not starting from scratch, but it is an earlier program than the next U01 we'll talk about. It includes screening for candidates, demonstration of relevant in vitro biological activity and target engagement, primary proof of concept efficacy data, as well as ADME tox and pharmacokinetic, pharmacodynamic data. So the year are September 2019, in March 2020, and the award is made in July of 2020. So it's often encouraged to submit a letter of intent for these applications so that we can get help our review team gather the best scientific expertise. Also, so the budget is may not be $300,000 in direct costs per year, and project period may not exceed 3 years. So this is a 3-year grant. No renewals. So one is for more advanced studies where a lead compound is already identified. So here is that at the end of the 4 to 5 year project, an optimized lead compound ready for advanced development activities, possibly funded by BARDA or another agency that leads regulatory approval and licensure. In this, it is recommended that you seek expertise in drug development and regulatory approval. This efficacy human relevant animal models that's consistent with the intended use in humans, PK studies with validated analytical, an acceptable safety margin and a lack of drug interactions, and development of stable synthesis and formulation. More specific criteria that, if met, should be eligible for consideration by BARDA or other advanced development entities, keeping in mind that completion of this work does not guarantee BARDA or any other entity will fund the advanced development of your candidate, but in theory, it should be ready for consideration for the next phase. The process should culminate with one lead compound with well-understood ADME, efficacy data and appropriate animal model relevant to civilian concept of operations, including the route of administration, and extended post-exposure treatment if necessary, if applicable for your candidate, and farm tox studies. It also have stable, scalable synthesis worked out and preliminary regulatory strategy, including a target product profile and a commercialization plan. There's clear that there is a preference for candidate therapeutics that have an additional clinical indication, in addition to the indication for chemical threats. And then finally, you should have intellectual property as well as, importantly, freedom to operate during the project period. So, competitive applications. We've learned that addressing each one of these criteria, perhaps in table form, is extremely helpful to the grant reviewers. A sample is presented here where you can see each is addressed, featuring the status, say it's complete or proposed, and where to find the proposed studies in the application. This reminds the reviewers what to look for, but also tells them where to readily find it in a long, complex application. So, general rule is to make application informative and easy to read. The Q01 program, the receipt dates are the same for the other one. The difference for this program is that the budget and project period is larger. The budget may not exceed $500,000 in direct costs per year, and the project period, instead of being 3, is a full 5 years. So we also do support large multi-component research centers of excellence. The first two to consider for these applications is, 1, we have strong preliminary data, and 2, if you have a team of researchers with projects that fit together synergistically around a central theme. Structure includes 1 administrative core for center management and operations, 3 to 5 interrelated research and development projects, up to scientific cores if needed and well justified, and 1 research education core. And the education core should be budgeted at no less than $50,000 a year and focus on the overall goal of increasing the number and capabilities of medical countermeasure researchers by building on the strengths of the center investigators. It has program oversight internally with a CounterACT Center steering committee, and externally with an external advisory committee that meets every year. These center projects encompasses basic research described in the R21 program through that described in the lead education and optimization UO1 programs. Her specific outcomes must be met depending on the type of application. At a minimum, you should identify a lead compound by the end of the project period, defined and described in the lead identification funding estimate. For all applications, you should have at least 1 optimized lead candidate as described in the lead optimization program. And second or more renewals, a clear track record of developing products that were transitioned to advanced development during previous project periods must be established. Note here that a table for these could work well here as well. Receipts are the same for the UO1 programs, and the difference for this program is that the budget is much larger, but the project period is the same. The budget may not exceed $2.5 million in direct costs per year and a 5-year project period.
At NIH needs special attention. Please read the specific notices listed here on enhancing and implementing rigor and transparency in NIH-funded research. Pay attention to describing scientific premise, specifically the quality, including strengths and weaknesses of the evidence from your lab and the literature. Be sure to address sex as a biological variable. If you do not use both sexes or you see no need to, have a good explanation why not. Differences is usually not an acceptable reason. Be sure to address biohazards, containment and disposal of, as well as authentication of key agents used in your research, including the chemical threat agent. Purchase something from Sigma or other supplier is not sufficient. You need to run an analysis to verify the identity and purity of key compounds. The emphasis here is on key agent and does not include minor reagents and buffers. Use the vertebrate animal section for experimental details to circumvent page limits. Viewers are not required to consider this section for that purpose. It may result in less enthusiastic review scores.
Important CounterACT specific issues to consider are how the therapeutic you are proposing will be used in humans. Especially important for the more advanced studies in the optimization and centers programs. If you see no real-world utility, the rest of the review usually does not go well. We also research with vulnerable populations such as pediatrics and pregnant women, and also encourage working with drugs that are already approved for other indications. Sometimes, and that's sometimes, the regulatory pathway to approval is less difficult. Therapeutics that can be used both as a medical countermeasure and a more traditional indication are a priority. For example, anticonvulsant used for nerve agent-induced seizures as well as epilepsy would be desirable, since the market for such a therapeutic would be much larger than one that treats nerve agent exposures. Strategy: include the structure of your compound when possible. Look at FDA guidances for help with regulatory strategy. FDA animal rule guidance that can be found on our website. They are used for all our programs except the R21, based on results or outcomes, not simply that a task or study has been completed. We strongly advise you to look at our website for instructions on how to write effective milestones for these programs. For property, the goal is to show that you have freedom to operate during the project period and beyond, and a commercialization plan is also required for some of the funding announcements. You should seek help from experienced collaborators.
Presentation part of the webinar. We would like to thank you all for taking the time to listen and hope it has been helpful to potential grant applicants. There is a list of NIH staff that participate in the program. Their contact information is available on the CounterACT website listed here. Please feel free to contact any of us for one-on-one consultation. We will now address some specific questions we have received.
Okay, just to follow up on that, when I will be sending out these slides by the end of this week. The recording to have it transcribed, that'll take a little bit longer, but you can definitely expect to have the slides sent to you later this week. And I will make sure to include Dr. Jett and Dr. Spriggs' email address on there, because we do advise you to reach out if you have any questions. So without further ado, let's get started with these questions. If just bear with me 1 second. First question. There's a question about how UO1 PIs and co-PIs are recruited or organized as a team.
Well, I guess as you know, we don't, the way we recruit people into the program is just by advertising the funding announcements. The teams, in terms of the teams, I would just, what you'd like to do and what you would need to accomplish that goal, and then find collaborators that could add value to the project. What I would advise is if you want to see who's already funded in the program, go to our website and we list all of our funded investigators. We list the title of their project, and if you click on that title, it takes you to the NIH Reporter, which is a database that has just lots of information on that project, from who's involved, what they're doing, and might get an idea of some potential collaborations.
Okay, next question. Can we propose experiments that will be done by the internal CounterACT team?
Best answer is we don't have an internal CounterACT team. This is an intramural program. Our labs are not working with us on this. They've got some other stuff to do, but they're not working with on this. So I guess the answer is, we don't do any research internally.
All right, next question. I would like to learn about the range of drugs being funded and how to collaborate with Centers for Excellence.
Okay, two pieces. So the first one was of drugs, candidates, not the threats, is from what I understand. And in that case, remember, this is a long list of many different kinds of chemicals that have many different kinds of effects. So it's just pretty large. I mean, we can go from anticonvulsants to antioxidants to anti-inflammatory drugs to drugs that sequester the agent, agent base, not host base, and on. So the answer is, if you wanted to look if you wanted to sort of look what we're doing, that's not to say that we're confined by that, but if you want to look what we're doing and see the kinds of different drugs that are being developed, a website might be a good place to start.
Next, what priority chemical threats identified by agencies? Yeah,
so as you've got a couple of slides, I'm not sure what the numbers are, but if you go back through our webinar, you can a couple of slides that address that issue. Now, you're talking about if you're looking about priority in terms of the prioritized list of chemical threat agents, obviously that's a classified document. But again, the main thing to do is to just give us a call. Let us know what you're thinking about. Let us know what the kinds of chemicals you're thinking about, and we're glad to help you to see if it would be feasible to put together an application.
Okay, next question. To what extent do projects need to be developed for competitive application in the R21 and R01 formats?
So R01s, we don't support R01s. Ours are U01s. A lot of that was covered, I think, in some covered in the presentation. But I'll say this: for R21s, typically they do not require a lot of preliminary data. In some cases, any preliminary data. I always tell people that, well, two things: there's a pros and cons of putting in preliminary data. So the pros is you can make a better argument for scientific feasibility when you have preliminary data. Cons are, if you put preliminary data in there, it'd be reviewed by the reviewers, and if it's weak, it could your score. So it's a cost. Now, I say this: if for R21s, I'm speaking just for R21s, if you can make a really strong argument for scientific feasibility from the literature, and remember, when you put that literature in there, you want about the quality of that literature that's putting in there. This study was really good because it was blinded, and had a bunch of rigor reproducibility. This study, suggests something, but it's not quite up to par in terms of rigor and reproducibility. So, but again, if you can make a really good case with the scientific literature, it may be okay. For 201s, you need preliminary data. You need preliminary data as well as, if possible, data from the literature to support your scientific premise for those medicines.
Okay. How many times can you resubmit an application that was not funded?
Once. By NIH policy, you have on the initial application and then the one revised application. And submit another grant as a new application. It just won't be, you won't have a chance to put in your introduction where you address the summary statement.
Okay. Considerations for young investigators?
You know that this is one of my passions. Diversity young investigators is one of my passions. Now, the NIH has specific policies to increase investigators, the biomedical research community, but it's only for, at least at our institute, it's only for, and I think other institutes as well, it's only for percentiled applications. Our applications are just scored, they're not percentile. But I'll say this, if application that is on bubble or close to being on a line but not quite close, and you make an argument for that, you can make several different arguments like, well, this is a really important chemical threat, and we don't have any other thing in our portfolio, and this, that. But I always include the fact the applicant is a young investigator, diverse investigator, things like that. So we do considerations, but not the formal ones you see with percentile applications.
Which mechanism should small companies apply under to develop a CounterACT therapeutic and to maximize chances of funding success?
First part easily, and that's all of them. Obviously, the SBIR program, which we do participate in. If you go into the Omnibus SBIR funding announcement, you'll see it's down to just a couple sentences now, but at one point we had a whole paragraph about CounterACT in there. So you come through the SBIR. You can also come through any of those programs that we mentioned earlier, the R21s, U01s, I think even the centers, I think maybe, accept applications from so the second part was
develop CounterACT therapeutic and to maximize chances of funding success.
Oh, I see. Oh, well, that's hard. Well, I mean, it's harder or not. An SBIR is more likely than a CounterACT. Both of those are scored applications, rely on the scores. It depends on funding. I mean, sometimes the SBIR has a lot of funds to support a lot of grants, and other times they don't, and the same goes for CounterACT as well. So I don't really know how to address that, but be happy to talk to that person on a one-on-one basis to try to strategize.
Next question. Are opioids toxic effects on brain related to this call, or?
Yeah. Well, yes, in the sense that we have a program looking at overdose. We have a notice that was, if you go to our website, you can click on the notice where we talk about the kinds of interests we have in consultation with Spal and NIDA at National Institute on Drug Abuse. And the difference here is that we're in countermeasures that could be used after one of these toxic opioids or fentanyls used in a mass casualty event, not really just a single oath that's covered by other programs at NIH. But yeah.
Are oxide and fluoroacetate agents of interest for medical countermeasure development?
Yeah, they're on the list if that's what you're asking. And I don't know if I mentioned this earlier, but we are permitted to submit an application as long as it's on that list of chemicals. Whether in the final analysis, when we're deciding whether or not to fund your application, we're guaranteed, in many cases, we have to look at our portfolio and look at the distribution of different chemical agents within our portfolio. And if you have, a couple of applications with the same score, and one is on a working with an agent that is of higher interest to the institute, that one probably would be selected. But as long as you're on that list, your one application, and we rarely turn down applications because of the chemical selected. Very rarely.
Question. How possible are CounterACT funding opportunities in terms of what you're able to research once you get the grant? Does it vary with different grant types?
Well, we're not flexible. So by NIH policy, you have to research that has been proposed, reviewed, approved by the NIH. We can't give you the money and then you start working on something else.
Does it need to have facilities capable of handling nerve agents internally, or an opportunity to collaborate with NIH facilities to conduct these studies? If so, how is this budgeted?
Great question, common question. So, chemicals that we study are in fact scheduled chemicals, not Schedule I drugs, but in terms of the Organization for the Prohibited Chemical Weapons, OPCW, are on the Schedule I list, and they not be used, or they can only be used in approved facility, DOD facilities or DOD-supported facilities. And those include things like the nerve gases, sarin and VX, and mustard, and some drawing up. But those chemical weapons, they have to be done in collaboration with the DOD or a DOD-supported laboratory. And in terms of collaborating with them, the easiest way is to look. It's only a handful. I'm not talking about 20 or 30. I'm talking about 3 or 4. So you can go to our website and look at who we're collaborating with that are working with these Schedule I agents, and explore the possibility of collaborations with them.
Excellent. Do you also consider biological threats like viruses?
Really consider them a threat. NIH has 3 programs, biodefense programs, basically. There is a very large program at NIAID focused specifically on biological threats. Focus here is on chemical threats only.
Next, I touched on this about the small businesses. We can skip this one?
Yeah.
Okay. Next, are treatment strategies viable for R21 applications? Also, are in vivo verifications required for R21 applications?
Question, pre-treatment strategies. So we do recognize that treatments could be valuable, especially in the military, into theater where they're potentially use chemical weapons. They've used pre-treatments for years. Our focus in the civilian sector is a bit different, where we can really predict where one of these attacks or accidents are going to happen. So our focus is on pre-treatment and more on treatment therapeutics. With that, what we tell folks is that we accept applications on pre-treatments as long as it's a small, or as long as it's a component of a project proposal that includes post-treatment. So an application that comes in that's just pre-treatment is not to be responsive to these FOAs.
Next, is a special study section used for the R21 applications, or are standing study sections used for review?
All CounterACT applications, R21s, UO1s, U54s, are reviewed by a special emphasis panel that has done an unbelievably remarkable job since 2006, in putting together the, and you can imagine a lot of different kinds of expertise that are needed for these kinds of applications. It's not done by standing study sections. It's done by a special emphasis panel.
Okay, this next question, I just want to let you know this is the last question we have as of now. So if you do have another question, please type it in now. You can get a chance to go to the front of the line. So with that being said, let's ask this question. Can you address the success rate for R21 and UO1 applications?
This might be a quick answer because I don't have that data in front of me. I would say it's on par with the translational research programs at our institute, better, and it's probably on par with the SBIR program as well. So it's better than your, usual one pool. But are reviewed under a lot of scrutiny, a lot of different pieces that the reviewers have to consider. But yes, it's a little bit better. R21's, one thing about our R21 program that we're extremely proud of is the transition rate from an R21 to UO1 is one of the highest, if not the highest, across all of NIH. So we're really pleased about that. Hopefully that will continue.
All right, we'll get another question. Are in vivo verifications required for R21 applications?
I think it's not required, but if a strong argument for a UO1, it would have some in vivo verification, but I think that it's required. I don't, Shardell may not know that.
No, you're right.
Yeah.
Okay.
The reason for
Are relate to a wide number of threats considered more attractive?
That's another great question. Great in the sense that it sort of depends. The general answer is yes, absolutely. As you know, we created these toxidromes because we wanted to, and we have some that are early along and that some of are already in advanced development where that would be the goal. As you may not know, one of the goals of this program, the NIH BARDA programs, is to put a strategic national stockpile so that we stand ready and have our medical response capabilities improved. If you had, the one drug, and the one drug could add nerve agents and open, that's more cost effective than having one for each one of those chemical threats. So you're more attractive. Now, the only caveat would be if you're working with, let's say, 5 different chemical threats in one, trying to coordinate that trying to have enough time and budget do rigorous for each one of those can be difficult. Finally, when you have a drug that you're taking to the FDA, usually you can contact the FDA to verify this or not. Usually, you have to have data for all of those threats. But the answer to that is yes, a resounding yes. It's much more attractive, especially to advanced developers who are taking these to licensure like BARDA. And I'd recommend you to the website as well, so you confirm or correct what I said.
Are there any active applications, close clinical trial for synthetic opioid prophylaxis and reversal drugs at this time?
I don't know the answer to that, and I probably would have to defer to our NIDA colleagues. We have to refer to our NIDA to be able to answer that. So I'd be happy to do that, but I don't know the answer to that right now.
So the person that asked that question, if you could reach out to us, and we will make sure to direct you to the right person. With that being said, it looks like we've come to the end of our webinar. I'd like to thank everyone for joining us. Please do reach out and let us know if you have any suggestions on how we can make these better. We're always looking for tips and suggestions. You can expect these slides in your inbox by the end of this week. In the recording, like I said, we do plan eventually to have that on our website, and we will also send it out via Twitter, and we'll even add it to our listserv. So with that being said, I'd like to, well, we have one more.
Oh, yeah, I just wanted to say thank you to Tim Lydon for running this webinar. Great job.
Thank you very much. So we, till next time, thank you very much, everyone. Take care.